Hello, I'm Ricardo and I'm going to talk today about how we develop a visualization tool for haplotype blocks for wheat research and breeding. I joined the effort of the 10+ wheat genome projects to compare the different assemblies produced. We started with a single reference genome assembled in pseudomolecules. Researchers doing wheat improvement across the world found that the original reference was insufficient. The 10+ Genomes project was formed to coordinate the international wheat resequencing efforts. Using a similar method to the Chinese Spring, the initial assembly, 9 lines were selected for their diversity and breeding importance. This plot is showing the assembly length of chromosome 2A across different lines, and will be the base of the haplotype visualization. Additionally, 5 varieties were included in the project with lower continuity. The assemblies were not at the pseudomolecule level. However, this is enough to project the Chinese Spring genes and use the projections to roughly estimate the location of the scaffolds. In total, we have access to 15 assemblies with different contiguity levels. For the assemblies at the scaffold level, the chromosome length is artificially the same as Chinese Spring. As the assemblies have different qualities, we have to develop a method to be able to compare the coordinate systems between all of them. In this example, we want to convert the coordinates of regions R1 and R2 to a target assembly. We start with the original annotation, RevSeq 1.1. Then, we have the projected genes in the target T annotation. Since we know from which region each gene came, we can label the regions. However, we can already see that the region T1'3 is broken by T2'1. We can further use the assumption that the order of the genes must be conserved. This allows us to remove the T1'3 region. We can also see a possible insertion labeled as B. Since the order of the gene models is conserved before and after the B region, we can assume that those blocks are correct. We can't ensure that the block B is part of the original region, as it could be a real insertion or a misassembly. We can use this method to project corresponding regions across the different assemblies. Scientists improving wheat are interested in identifying regions that are conserved to be able to increase the diversity of the crops we cultivate. In this example, we identified a region that is the same in harina and maize. We also have some regions that are common in more than one assembly. We use different colors to identify different haplotypes. Once we add the information of all the haplotypes in a single plot, the interpretation is harder. For that reason, we developed an interactive view of the data. The initial view of this plot is the same as before. However, to help with the interpretation of the results, a table with the coordinates of each block is displayed. With this view, the user can focus on the blocks they may be interested. In this example, the user has selected the position around 169 million from the point of view of harina. The corresponding block in Robidus is also selected. The user can select a different region. In this example, the user has selected Cadenza. The identification of these long-conserved regions across the different wheat varieties is very important for crop improvement. This is because we are seeing a lack of diversity in this chromosome. Therefore, introducing variation targeting this chromosome can lead to new varieties. Besides the static view, the user can change the selection with the mouse. This is a zoom region where moving the cursor shows that in some cases the corresponding coordinates are not moving in the same order. The change in direction means that the region contains an inversion. Because both varieties with the inversion have the same haplotype, it is very likely that this is a recent event. Having the coordinates of the haplotypes is interesting by itself. However, researchers may be interested in a particular gene or genetic marker. To make this more useful, we have a search feature to find the coordinate position on each of the assemblies. This will highlight all the haplotype blocks that are under the gene. In this example, there are 3 different haplotype blocks under the selected genes: the green haplotype of harina and Jagger, the light orange haplotype of maize and Stanley, and the dark orange which covers most of Cadenza. However, there are a couple of red blocks at the beginning and the end of Jagger suggesting that the haplotype block has been broken in two instances, one Sinclair and the other in Lancer. We can keep adding genes to the visualization, and this will add the relevant haplotype blocks. In this example, the next gene shows that the original block from Marina has a small region in Mace and Stanley. Adding a third gene adds more haplotypes to the plot, as expected. On this example, we can see that the haplotype block in Mace and Stanley have the same haplotype in these three genes, but with a region that is broken. Furthermore, the second gene seems to be coming from the same source as Arena. To explore in more detail, the user can select a variety. If we select Stanley, we can see that the middle gene is very close to the haplotype coming from Arena, but it is removed from the visualization because it is not that the two green blocks are common between Stanley and Mace. Those are two independent events that have different coordinates based on the similarities to Arena. When we focus the analysis on MATIS, we can see that across these three genes, there are 5 lines in total that have a shared block. However, only in Paragon and Cadenza, the large block is conserved. In Claire, Jagger, and Lancer, we can see that there are some recombination events that sometimes cover the first gene, and sometimes it doesn't cover the gene, like in the case of Lancer, where the two genes are in a different block. And if we look from the perspective of Jagger, we can see that there is a very large region that has blocks across the different genomes, but again, they have different recombination events. I would like to thank the 10+ Genomes project for the early access to the assemblies that we used in this project, and in particular to Jemima Rayton, who worked with me in the development of the algorithms and the visualization, and in general the whole WOWI lab for the feedback and being the testing users who, of course, broke the site several times. And to you for your attention. I'm open for questions.